大家好，我係今日嘅司儀 Iris， 歡迎大家出席由香港科學館、香港中文大學新亞書院及生命科學學院聯合主辦嘅任國榮先生生命科學講座。除咗喺香港科學館嘅現場講座會同時於 Zoom、香港科學館 Facebook page 同埋 YouTube channel 進行直播。任國榮先生生命科學講座系列創立於2015年，由香港中文大學新亞書院及生命科學學院合辦，旨在令社會大眾同埋學生加深對生命科學嘅認識，以及生命科學喺現今社會嘅價值同埋應用。今次講座，我哋有幸邀請到台灣中央研究院生物化學研究所客座講座蔡明道教授為大家主講，係蛋白質的微觀世界。講座會以英語進行。我哋亦都邀請到香港中文大學生物化學課程主任邵鵬柱教授主持講座。講座完結之前呢，我哋會有問答環節。如果大家有任何問題呢，歡迎屆時舉手發問。收睇網上直播嘅觀眾亦都可以留言發問。我哋會因應時間代為向蔡教授發問。事不宜遲，我哋即刻開始。有請邵教授。
Okay, good afternoon, ladies and gentlemen. I have pleasure to introduce Professor Tsai to you. Professor Tsai received his bachelor's degree in chemistry from the National Taiwan University. And he received a PhD degree in medicinal chemistry from Purdue University. He was then a faculty member in the Department of Chemistry and Biochemistry at the Ohio State Universities in 1981 to 2006. Subsequently, he moved to the Institute of Biological Chemistry of Academia Sinica, Taiwan. And he also served as director during 2006 to 2014. He is also a professor at the Institute of Biochemical Sciences, National Taiwan University. He is now also a fellow of the American Association for the Advancement of Science, Academia Sinica, and also fellow the World Academy of Science. Professor Tsai's interest is on the mechanisms and also the functions of proteins. He in fact used a lot of different advanced and emerging techniques to probe these problems. Professor Tsai has trained over 60 PhD students and over 40 postdoctoral fellows, and some of them have become highly successful researchers. In addition, he has been actively involved in developing infrastructures and services, both in Ohio State University and also in Taiwan. Without further ado, let's welcome Professor Tsai.
Thank you, Professor John. And it is my great honor to have this opportunity to present this important lecture, particularly in the post-pandemic time, I think what happened with this virus has to do with proteins, not just viruses. So my lecture will be divided into 4 sections. The first is the basic knowledge about proteins, and then the protein structures at the atomic level and protein related to COVID-19, and then more advanced section will be watching enzymes in action. So I hope everyone, whether you are expert or non-expert, whether you are a scientist or non-scientist, or whether you are an adult or young children, kids, you get out of this 1 hour. So this is what we know. Eggs are rich in proteins. So we want eggs to be cooked for breakfast. Now, the cooked eggs, the proteins, they have a lot of proteins. The proteins are denatured. Okay, so you eat the proteins, and they are digested in your body to produce energy, and then some essential amino acids, and then they are used. The energy and the amino acids are used to synthesize the proteins that you need for your body. If the egg is hatched, then a little chick will be produced. And the proteins are used for the nutrition of the embryo. Okay. And in a research environment, what we are more interested in is an uncooked egg where proteins retain the structure and the activity, which can be purified and characterized. Okay. So not only eggs have proteins, the proteins are everywhere. Okay. They are involved in almost every function of the body or the organism, particularly also in the infection of the SARS-CoV-2. Many of the proteins are also enzymes. Okay. Enzymes catalyze reactions that happen in our body. Okay. Proteins could be it could be muscle. Muscle has proteins. Is a structural protein. Antibodies, we talk a lot about antibodies in the fight against COVID-19. The antibodies are proteins. They are important proteins. Okay. So we don't have to go through all of these. But in the basics, where do proteins come from? Okay. What is more important? Are proteins more important or genes more important? Okay. Genes are important. DNA is important because it carries the information, genetic information. But the DNA has been transcribed into RNA, and then RNA translated into proteins. And proteins do all the functions. So I would say protein is more important, and protein is also troublemakers. Okay. If some protein goes wrong, you got a problem. Okay. If some DNA goes wrong, you got a problem also. Okay. So the messenger RNA vaccine that everyone knows for COVID-19, it uses the same principle. That is, you take a piece of viral RNA and then you inject into our body, and then our body has this translation system to produce a small piece of viral protein, which then stimulates the production of antibody in our body, which then fight against or defend us against the viral infection. So there are 22 amino acids in proteins, and 20 of them are the most common ones. Okay. And I'm sure you have seen the chemical structures of each of these amino acids. Okay. Only 20 some amino acids, and each of them only has 3 or 4 different kinds of atoms: carbon, hydrogen, and oxygen, and sulfur, and selenium, and nitrogen. Okay. But they make up proteins to do important functions. And this is the ribosome, the schematic illustration of the production of proteins that occur in the ribosome. And this is the messenger RNA, and then the amino acid carried by transport RNA, and then the protein is generated here. So this is the primary sequence, which simply one amino acid is connected to the next amino acid. Okay. The proteins that is first synthesized is not folded, but usually it is folded spontaneously in your cells, okay, into what we call a tertiary structure. Okay. And the folded protein would have functions. Okay. Unfolded protein may also have functions. Yeah. Now, between the primary structure and tertiary structure, there are secondary structures like alpha helix here, and then beta sheet here. And these structures, they are composed of, or they consist of hydrogen bondings or some hydrophobic interactions, or and so on. So in other words, the chemical principle of these structures are actually very simple, something you learn in middle school or high school about basic chemistry. That could tell you cover all the bits of proteins or protein structures. And this is a one way of representing a protein. That is using the ball and stick model. That is, every atom is the ball, and then every bond is a stick. And one way of looking at it. Okay. Another way of looking at it, a protein is a cartoon model. Okay. So you would use a ribbon to represent secondary structures. Okay. And then the other way is also surface representation. Because amino acids have charges, so the surface of protein have different charges or different combination of charges. So you could use blue color to represent positive charge, red color to represent negative charge. And this is important because proteins need to interact with other proteins. Okay. Proteins don't just work by itself in most of the time. Okay. So when it interacts with other people, then other molecules, then the charge is important. So this is a animation of a protein just so that you could see the three-dimensional structure view of the protein better. And this is a DNA polymerase from African swine fever virus. Okay, that is a virus that we are familiar with. Okay. And this enzyme, so it is a polymerase, so it is an enzyme. In this case, in this structure, it binds a nucleotide at its active site. So usually an enzyme structure has some spaces to bind its ligands. Okay. And this is this on the left is the same enzyme, but in addition to the nucleotide, it also has DNA bound, okay, because this is a DNA polymerase. It needs to replicate DNA, so it needs to bind DNA. And the right is the same, also a DNA polymerase, but it's from human, okay, it's a human enzyme. And the difference, as you could see, is the human enzyme is larger and actually has an extra domain. Yeah. So you could have the same enzyme, the same protein with the same name, and maybe doing the same function, but if they come from a virus or come from bacteria or come from human, then they could have a different structure, or at least somewhat different structure. And this is what we are interested in. That is, why are they different, and what the difference makes. So I'll give you some idea. We know proteins are very small because we are talking about microscopic world of proteins today, right? So what is shown here is a relatively large protein. As you could see, it has 12, actually have 12 subunits or polymers. The diameter of this protein is about 5 nanometers. Okay. So how small is 5 nanometers? Can you imagine? So for comparison, I'm showing the largest object that could be observed, that has been observed. That is a black hole, not directly, not by eye, but by microscope. And then for comparison, give you some idea. There's Earth, and then there is an apple. The diameter of apple is about 5 centimeters. So what is the difference between an apple and this protein molecule? The difference in the diameter is 10 to the 7th fold in terms of diameter. And in terms of volume, then you need to take another power of 3. So that's a huge, huge number. Okay. But then if you want to give some idea, then you could imagine what is the difference between an apple and the Earth. Okay. That is even greater by an order of magnitude, that is, or 3 orders of magnitude. That's 10 to the 8th in diameter. Okay. And then for the black hole, it's another factor of 10 here in the diameter. This is all approximate. Okay. So that is why we could not see the black hole. We have to use telescope, right? So we could not see the protein either. We have to use some kind of microscope. Okay, some very, very advanced microscope. Okay, so this brings us into the next section. That is the protein structures at the atomic level. Okay, now we have to think small. Don't think big anymore. Okay. The atomic resolution of proteins is in the order of angstrom. Okay. Angstrom is 1 nanometer. So why do we need this information? Because we need to understand the specificity of proteins or the specificity of biology, because proteins do the work in biology, right? Now, biology is very specific, right? You are you, I'm me, okay? We are all human beings, but we are a bit different. Okay? We are not identical. Okay? So everything has specificity. An enzyme has specificity. The enzyme specifically contribute to the specificity in biological functions, but it is the structure of the enzyme that contribute to the specificity of enzymes. So chemical basis of protein functions. The design of new inhibitors for drugs. Okay, many drugs are inhibitors of enzymes. Or even there are protein drugs that is becoming more common, as antibodies could be one type of protein drugs. So many Nobel Prizes have been awarded to the development of methods to solve the protein structures, or as well as the solution of important protein structures. Yeah. So if you if you have to solve the important structure of a protein, you have a chance to get a Nobel Prize. Okay, it seems not that hard. So I'm going to go through the three major methods to solve protein structures. And of course, this will not be in too much depth. This is just for your entertainment, hopefully. So the first one is X-ray crystallography. And there are some, at least a few experts here in this audience now. So the first Nobel Prize was given to, this field is to in 1962 to Perutz and Kenteru for solving the hemoglobin structure. Okay. And this is the cartoon model of hemoglobin, and it is based on crystals. Okay, so this theory behind this is the X-ray goes through crystals, and the X-ray beam is diffracted. So from the diffraction pattern, since the crystal is well ordered, so from the pattern, you could then solve the structure, okay, like the one in the lower right corner. That's a structure. So, and in the older time, this could be done in a laboratory. The instrument could be in any of the institutes in a university or research institute. I'm sure there were many in Hong Kong universities. But in the last 10, 20 years, this was done almost entirely in a synchrotron facility that usually is a regional or national facility. Now in Taiwan, there used to be a smaller synchrotron facility here. In the recent years, we built a newer and larger ring called Taiwan Photon Source, and that is one of the the brightest light source. DNA polymerase, I mentioned it already. The Nobel Prize was awarded to Arthur Kornberg for the discovery of this enzyme because it replicates DNA. And the structure was solved later in 1980s by Tom Stites. And channels, okay, what is channels? See, many things need to go through our cells, in and out of cells. Okay. So cells have membranes, and membranes have channels. Okay. There are water channels, there are potassium channels, there are sodium channels. There are many different types of channels, and they are all specific. Okay. The water channel would not let potassium go through. Potassium channel would not let water channel go through, even though they are similar, right? At this type of resolution, they all look similar, but details are very different. And that's what scientists are interested. And then this is the RNA polymerase. So RNA also needs to be replicated or to be transcribed. Okay. Transcription of DNA to RNA. And then ribosome is the place that RNA is translated into proteins. Okay. So these are cellular protein machineries. They are important. So the atomic Nobel Prize is awarded to this group's work. And more recently, signaling. So the cells have receptors which receive signal from outside and then tells what to do, molecule, what to do inside. And a very common and useful receptor is G-protein coupled receptors called GPCR. And this class of receptors also received a Nobel Prize. And the second major technique is nuclear magnetic resonance. And I'm going to be very brief on this. So a protein molecule has many atoms, right? Maybe hundreds of atoms or thousands of atoms. Even though most of these atoms are just carbon and nitrogen, but they all are in different chemical structures. They all have different chemical environment. So if they are placed in a magnetic field, then they will experience slightly different magnetic fields. So they will have slightly different shifts or resonances. So if you do NMR experiment, you could see many signals like this, and each of them could represent an atom. So there are hundreds of peaks here. You do hundreds of atoms. All you need to do is to identify them and learn their relationship from each other and correlate each of them with an atom in the sequence. Then you got the structure solved. Okay. So, and Nobel Prize was given to Richard Ernst for developing the methods of NMR, and then later another Nobel Prize was awarded for doing protein structures by NMR, as well as for development of mass spec. That is another important technique that I'm not going to get into today. Now, all of you are familiar with MRI, magnetic resonance imaging, which uses the same principle as NMR. Okay, so there was also a Nobel Prize awarded to MRI. So the more recent development, and which contributed a lot to the COVID-19 research, is cryo-EM, cryo-electron microscopy. And this was 2017 Nobel Prize that was awarded to this technique. And this is a technique that you don't need to rely on an X-ray or diffraction or NMR manganese. You are looking at the molecule directly, okay? Except that you use a microscope. So the theory actually is simple. Okay, so these are all protein molecules. Okay, they are the same protein molecules. Okay. Now, protein molecules is laid on the surface of something, a grid or something. Okay. Now, each protein, if they don't overlap, what you need to do is to make sure they don't overlap with each other. Then each protein will be laid at a random orientation, right? Not like all of us, we always stand, right? We don't have to all stand. You can lie around all the time randomly. Then if you shoot electron beams through, then there will be an image or shadows like this. Since the orientation is random, these images will also be random. So in principle, all of these random shapes, images, could be assembled by the computer and use a software program to solve or make up the tertiary structure. Okay. Only that you need enough sensitivity. Therefore, you need to have many thousands of images so that you could line up the same images to enhance the sensitivity. Okay. Otherwise, the principle is very simple. Okay. So when this breakthrough happened, well, actually this cryo-EM has been around for many years, but there was a breakthrough in the imaging technique and sampling technique also in about around 2015, and then it became highly feasible to do biological molecules. So we in Academia Sinica then also rushed to set up this cryo-EM facility in 2018, and then we had a grand opening and a workshop and learned how to do it. And then by 2018, we have produced this structure that I showed on the earlier slide. Just don't worry about what this protein does. Just enjoy the nice colors and shapes. So it is 12. It is a dodecamer. That means 12 units, subunits. Okay. So it is a hexamer of dimers. So there are 6 dimers. So this is the dimer. Okay, one is darker color, one is lighter color. Okay. And then each of the dimer, each monomer, is able to bind some ligands in the active site. So this is, NADH is a coenzyme, and then magnesium ions, and then these are the substrates. So what is shown is animation to show you the details. Okay. Okay, now let's talk about COVID-19 proteins. That is greatly facilitated by the development of cryo-EM. Okay, that happened just in time. Otherwise, we may have done much worse. So many of you know about this picture. Okay, that's we know the virus, and this is how the virus infects the host cell. So the viral surface has this spike protein. Okay, it has other proteins. Okay. The spike protein functions plays a major role in the infection. That is, the spike protein actually interacts or binds to a receptor or something, another protein on the surface of the host cell called ACE2. Okay. Now, it is with this binding that the virus could then break into the cell. Yeah. So the 2 proteins which are most important, one is the spike protein of the virus, the other is the ACE2 protein of the receptor. Okay, we want to know their structures at atomic resolution. But we have to start from the gene because fortunately, the sequence of this genome, this is the RNA genome for the virus, was available, published in the public as early as January of 2020. Okay, that's very, very fast. And from the genome sequence, we could know exactly what is the sequence of the spike protein. Okay. And then from the protein sequence, we could solve, we need the information of protein sequence to solve the protein structure. And then from the protein structure, we could learn about the infection mechanism. And then based on that, we could move to the drug development, or even development of antibodies. So all of these activities happened around the world, starting from January of 2020 until now. And it's even going now. So this is the structure of many of the, one of the many hundreds of structures that have been solved for spike proteins. And it is a trimer. Now, the trimer, however, are not identical. These are two of the three protomers of the trimer. And as you could see in this one, this part of the molecule is different from this part of the molecule. And this part of the molecule is called RBD. RBD is a receptor binding domain. That means the domain that binds to the ACE2 receptor. That is a region that interacts with the host cell. So this region is most important. And since these two protomers have somewhat different conformations, so the scientists, at least in this publication, they immediately proposed that this is important. This has to do with the easiness of infection, or the effectiveness of infection. And this structure was published in February of 2020. That's also very, very fast. And once you have the structure of the spike protein, then the next is to do the complex between the spike protein. So this is only the RBD part of the spike protein interacting with the ACE2 protein. So these two, they bind together or interact together, and the structure are solved. And not shown in detail here, but you could actually look into the data and look into the detailed information, detailed interaction between these two proteins. And that tells you how to design inhibitors, for example. And then you could also compare the complex, so this is the part of a small expanded part of this this region. You could compare with the SARS-CoV-2, the COVID with the SARS 20 years ago, and you could see there are some differences. So that helps scientists to understand what is the difference between the virus we have this time and the virus we have last time. Okay, we are not talking about details today, but this kind of activity is what happened very actively in the scientific community. And many of the papers were published around the world, but this paper, important one, came from laboratories from Beijing and Shanghai. And the next one is a comparison similar to the previous slide, but the comparison between Omicron and the Delta strain. Yeah. And this work was also published from the laboratories from different places in China, and one of the co-authors came from the University of Hong Kong. And then there are many antibodies developed for against the spike protein. So we want to know how the antibody binds to the spike protein. So this is the structure of an antibody binding to the RBD region of the spike protein. And again, if you zoom in the details, then that could tell you important information of how this antibody works. And if it is not working well enough, how would scientists be able to improve it? And one of the small molecule drug that was on the market very early on was remdesivir, remdesivir here, which is a inhibitor for the RNA-dependent RNA polymerase, which is responsible for the replication of viral RNA. Okay, so the strategy, you could have different strategies. One is to target the entry of the virus, or you could target the replication of the virus. This is one example. These are just two of the examples. There are many approaches or protease inhibitors. Okay. The final drug is based on protease inhibitor. So the final part, I will talk about watching enzymes in action. Okay, so this could be more for the advanced, but I hope I think you could understand it. So enzymes catalyze reactions or chemical reactions. Okay, many of the chemical reactions that happen in our body would not be possible without enzymes. Okay. That's very important. So what does enzyme do? What is the trick that enzyme do? Okay. Enzyme bind some substrate and then convert it to other substrate. Okay. So the enzyme reaction is actually a process. Okay. So the enzyme, so this is again a DNA polymerase. So there are 4 different structures here in 4 different colors, but let's start with the green color. Green color is E is the enzyme, just the enzyme, before it binds anything. So DNA is shown in this color. So this is a light blue color. This is DNA. So if you look at the green color of enzyme and the blue color DNA, that's one thing. But then it forms a complex. Okay, once DNA binds to the enzyme changes its structure somewhat. Okay, actually, we call it conformation. It does not, the structure is still the same, but the conformation is different. So it's called a conformational change. So now the pink color, okay, is somewhat different. Now, the next is it needs to bind the nucleotide. Okay, so this is the nucleotide. And then the enzyme become a blue color. Here is a blue color. So the blue color, again, is somewhat different from the pink color. And the DNA in this, in the presence of DNTP is also somewhat different. So we could look at these motions. So the motions are animated, not the real motion. Okay. Why are they animated? Because we only were able to take first structure, take second structure, take third structure. Okay. We prepare free enzyme, we prepare enzyme DNA complex, then we add the next nucleotide and so on. That's what we do, scientists do all the time. Yeah. But what we like to know, or in the next generation, or you could say that an emerging field is to watch the motion, okay, to watch the structure changes at real time. So the motions actually happen. Of course, the enzyme has to move to do something, but how fast does enzyme do? Okay. If the enzyme is very slow, then it's ineffective and we may not be able to survive. Okay. The enzyme catalysis, an enzyme molecule usually work about 10,000 cycles per second. Okay. I mean, this is very roughly, okay. That means from the beginning to the end and start over, usually take a millisecond or less than a millisecond. But a cycle consists of many steps. So every step will be much even faster than milliseconds. So the time scale of enzyme catalysis in milliseconds, around milliseconds, the chemical reaction could happen in nanoseconds to picosecond or even femtosecond. Okay. Some domain motions, ligand binding, protein folding, they have their time scale. We'd like to know those. The reason we are not doing as well as we should be in terms of developing drugs, in terms of fighting diseases. The main reason is because we don't know enzymes well enough. Okay, that is very clear to me, okay, but not very clear to many people. Okay. So how long is 1 nanosecond, for example? Think about it. But just when you think about it, the difference between 1 second and 1 nanosecond is about 30 years old to 1 second. So if you are 30 years old now, you think about just 1 second. Okay, that's 1 nanosecond versus 1 second. Yeah. Of course, that's a nanosecond, but here is picosecond. So this is very interesting to think how fast is fast, how slow is slow. Okay. So there is an emerging method called time-resolved X-ray free electron laser. That we could do time-dependent study at time scales from femtosecond to milliseconds. What is an X-ray free electron laser? It is X-ray. Okay, that is the same as X-ray, except that it is a laser X-ray. So the laser is much more. Okay, laser has high intensity and its pulse duration is much shorter. Okay, we all know this about laser. Okay, therefore, compared to the regular synchrotron X-ray, this free electron laser X-ray is much stronger and intense and focused. Therefore, allow you to do some special experiments. And this kind of facility now is available only in 3, 4, or 5 different places in the world. And the experiments that we did was performed in 2 places. One is at the Sakura facility, of Japan. And then the other is at Switzerland. And we started in 2017, just before the pandemic. Actually, we worked through the pandemic and only was interrupted by 1 year. And at that time, to travel, even to get into the lab, require special approval, of course. But some of the team members, they were very determined to pursue it. So the system or the problem we were trying to solve is the repair of DNA damage. So our DNA in our body are being damaged all the time. That happens very often. Okay, but sunlight could damage your DNA, for example. But damaged DNA could be repaired. So our body has enzymes to repair DNA. But if it's not prepared properly, then you got a problem. So this is an enzyme. This is a DNA damage where two thymine bases form a dimer. Okay, so these two thymine bases form a dimer. Okay, so it's called a cyclobutane-pyrimidine dimer. Okay. And it is damaged due to UV light. Then there is an enzyme called photolyase. This enzyme could repair this damaged TT dimer, but this repair is also triggered by light, by blue light. So what we want to do is to do a light-dependent, time-dependent structure to watch this process. And this slide looks complicated, but it is not so much. So first, this is a diffraction pattern that is the same as what I showed earlier. Okay, just diffraction pattern. The important thing here is we need a pump, we need a laser, a blue light to stimulate it, to initiate the enzyme, activate the enzyme, so that the enzyme could start the reaction. That's the blue laser. Then we need a X-ray laser to collect the data. So once the stimulation, the pump laser occurs to stimulate the reaction, then at certain interval of time, we shine the X-ray laser and then we collect diffraction and then we solve the structure. And this time interval could vary from T1 to T2 to T3. So the time interval could be very short. It could be nanosecond or femtosecond, only trick. Okay. Okay, so this is the result, and this is a time-dependent structure of the damaged DNA. The part of it, I'm only showing part of it for you, to see. So each structure here is a superposition of two structures. One is at the green structure is at time 0 before you shine light. Then the other color is at a different time. So this first one is at 250 picosecond. Second one at 150 picosecond. And then 650, and then 1 nanosecond. So as you can see, nothing happened at 450. At 450, something to be happening. There's slightly deviation here, but at 650, clearly one of the bond is broken because this distance becomes longer. And then at 1 nanosecond, both bonds are broken. And in other words, the ring is opened up at 6 and by 1 nanosecond. Okay. So in other words, you could say the chemical reaction occurs in 1 nanosecond. Okay. Now the enzyme did that. The enzyme helps. How did the enzyme? So we turn to the enzyme at the same. This is all from the same set of data from the same time point. So now we could look at the enzyme at 250 picosecond or 100 picosecond. This part is the CPD, the damaged DNA. If we look at these details, so again, let's focus on one thing at a time. So if we focus on this region, there are five balls at different colors, right? A green color and red color. So these represent 5 water molecules. Okay, why are they there? They are just part of the protein. Okay. So when the reaction starts, even though these 2 colors, they are very, very similar. They almost correspond to each other. But then these structures begin to break apart. Okay. And by 1 nanosecond, this is 1 nanosecond, the repair is complete. But the enzyme, okay, these 5 balls are still scattered. Okay, the 2 colors still don't superimpose. Sorry. So by by 500 nanosecond, the 5 water molecules, they come back, superimposed again. Okay. This is one place you look at it. You could look at another place. You could look at the arginine. This is another amino acid. At 100 picosecond, okay, a fraction of it has moved, and then it moves. But then it scatters around, and then at 500 nanosecond, it goes back. So the point is, chemical reaction occurs at 1 nanosecond, but the protein, the enzyme, recovers, takes 500 nanosecond. Okay. And you could look into the detail and look at the detailed steps. Okay, but at this time, the damaged DNA is still there. The repair DNA, okay, is still there. I'm not going to show you the next steps. This is too much detail. But after that, the repaired thymine will return to pair with the original partner. And that takes another, that will be in the microseconds range. Okay. So the best thing is to show you a motion picture here. Now, this is not animated. This is from the real data. Okay. So time is shown here. Okay. Different time here. And then different parts of the molecule. This is DNA. Oh, so 1 second, 1 nanosecond is complete. Okay, but then the protein is doing something. It needs to return. Okay, now the bases are returning. See the here. So this is not very sensitive. So okay, here. They return to pair with the original DNA. Yeah. And that is, remember, this is in the now 200 microsecond range. Okay. So 200 microsecond is more like the right time range for the enzyme to complete the cycle. Okay, that's what I mentioned earlier. Okay, but the key part, the chemical reaction occurs very, very fast. Now, why do we want this kind of information? Because if we know such information, then it is easier, better for us, or possible for us to design inhibitors. Okay. I mean, this is just an example, but many diseases are caused by enzymes, and we need to design inhibitors or do something else. So we need to know the proteins, enzymes at the right time scale, at the good, high enough resolution, and then we will be able to fight diseases or develop drugs for the future. Okay, AI is very useful to help us, but we have to do what AI could not do. These are the things that AI could not do. And there are plenty of things that AI could not do. So from the conventional structural biology, I use this as a comparison. That is, if you take a nice picture of the symphony playing, you take a snapshot, but you don't know what they are playing, because the music is a process, just like the enzyme catalysis, the process. And you could imagine this is a nice structure, and each player, each instrument is like an amino acid. So what we want is to know the extent that we could hear the music like a real-time catalysis orchestrated by an enzyme. Thank you.
教授帶我哋進入蛋白質嘅微觀世界，加深我哋對蛋白質嘅認識。而家進入問答環節，有請邵教授、蔡教授到台前就座。歡迎現場觀眾舉手發問，請盡量以英語或普通話進行提問，而網上嘅觀眾亦都可以留言發問。而家將時間交俾邵教授、蔡教授。
It's a very exciting talk. So, for the audience, yes, please.
How can the proteins known as antibodies help to fight disease?
Okay, how the antibody can be used to fight disease.
So antibody is specific in binding to a specific target. So if, for example, the spike protein has to find the receptor, right? But if you have an antibody that binds the spike protein, then the spike protein could no longer find the receptor. That's how it works.
Okay, yes, please.
If the human body's dead, do the enzymes or something still moves?
He just asked how this protein robot recognize the substrate. Is there any intentively find the target or just a random reaction?
That is actually a very good question also. The antibody one is also a very good question. That is, how does enzyme find the substrate? There is different theories. It could be random, okay, but it's not random. Okay, there are different theories. Okay. One theory is it's totally random. If you have enzyme and substrate, assuming not in the body, you put it in solution, then they are all in motion and they find each other. They happen to hit each other, then they bind. But once they bind, then the equilibrium may shift. Okay, then one step forward. But this is one theory. The other theory is there is enrichment. You know, in cells, there are compartmentations. That is, this enzyme and this substrate they are located nearby each other. So they are easier to find each other. And there is even a third situation where we sometimes call it tunneling. That is, some enzymes may form a complex. Okay. So maybe the product of enzyme A is the substrate of enzyme B, but enzyme A and B already form a complex. So the product from enzyme A is ready to bind enzyme B. Okay, you understand? Very good question.
Any other questions? There is a question from the Zoom. So can you discuss any challenges or limitations in observing protein molecules at nanometer scale?
Yes. So again, we don't use our eye to observe directly at no scale, but we use a microscope. Now, microscope is the helper. So then we assist it by computer to put all the images together. But since microscope is an instrument, that is a microscope, you look into it. So in principle, we say we could almost watch it directly, but in effect, it is not. Okay. But in terms of challenges to do this kind of experiment, I think this is a very common experiment now in many universities. But if you are talking about experimental challenges, then I think the first challenge is sample preparation. Okay. And you need to spread samples, and sample in a very thin condition. That is, molecules don't overlap with each other, because if they overlap, then they cover each other. You don't know the shape of the molecule. Okay, that's one challenge. There are other challenges, but if you have a deeper question, you could ask.
Okay. There's another interesting questions from the internet. So can we recover or reactivate a delayed proteins, such as a cooked egg?
Okay, could you repeat again?
Can you recover a delayed proteins? Yeah, such as a cooked egg. Can you make it become a raw eggs again?
Yeah, in general, you could. You mean denatured protein in general? Which particular protein?
Just in general.
In general, yeah. In nature, in general, some proteins, at least all proteins, could be denatured, renatured. Okay.
Yeah.
And
how about a mixture of proteins like an egg? Can you cook it and then make it become a product?
No, egg is better to be just eaten.
Okay. Okay. Yeah. Okay. Any other questions? Yes, please. Just wait for a moment at the microphone.
Yeah, thanks, Professor. A very nice speech. I'd like to ask is that I think that you use the blue light to trigger the DNA repair enzyme. And what if in our bodies, I'm not sure whether it's also a light trigger repairing process.
So the enzyme I talk about does not exist in our body. Okay, it is a plant enzyme. Yeah, but in our body, DNA repair uses a different mechanism. There are different mechanisms of DNA repair. Not by light triggering, but the light could cause DNA damage to our body.
Okay. There's another question from the internet. So how do you see AI technology contributing to the study of protein molecules? And what specific applications or advancements do you anticipate in the field?
So AI has already been used in developing structure prediction programs. So you could give AI as something like AlphaFold, a sequence, and then you will get a structure back. Okay. Now, at this stage is how well does the prediction into the compare with the experimental structure. Okay. So for now, we would not trust what AI could tell us. We could use it as a reference. So if you want to solve a problem, if you have a protein that you don't have structure, no matter how you try, you could not get a structure, then you could use AI to predict. And that would be, you could call it a model or a hypothesis. Okay. That is useful. And that could help us to guide us. AI could tell us the global structure, for example, but not in the high resolution. What is important is what matters in life is not big things, some small things. When a protein matters, when a drug works, the trick is usually in very fine detail. In the scale of angstroms, or if maybe a couple of angstroms. Okay. So that is the gap we are facing now.
Okay. Any question from the audience? So we asked about enzymes. So different body parts have different type of enzymes, and also can we increase our enzymes in the body?
Can we increase our enzymes in the body?
By eating those pills?
Well, okay. There is, if you are talking about disease curing, then there are gene therapy, for example, or even protein therapy. I don't know. There's cell therapy. I think you could do almost anything you want to our body now. I think the issue facing us is not what you could do, but what you should not do. That would be more of a problem, including AI. What should AI do? What should AI not do? That would be more of a problem. But if you do more exercise, I think your body will have more enzymes.
Okay. So he also follow up that would the enzymes and the protein in the body be used up someday?
No. As long as you are alive, it may be out of balance, but it would not be used up. Also, some bad enzymes may happen. If it's out of balance, then it could be a problem.
Okay. Any other questions? Yeah. There's one here. He talked about how to study the detailed three-dimensional structures of a protein. So do we need to unfold the protein before we study the detailed three-dimensional structures?
No, you don't need to unfold the protein to do that. Studying protein unfolding is also an important subject, and it also is related to neurodegenerative disease. And so that's important to understand how protein folding and unfolding occurs. Yeah. But to do the structures that I talk about, you just use folded protein. Unfolded protein will be harder to do.
Okay, any other questions? Yes, please. There's one there.
What led you to study proteins?
Could you repeat?
What led you to study proteins?
To-
What makes you to study proteins?
Why do you study proteins?
Why do I study proteins? Okay, that's a good question. Well, I study chemistry. I happen to study chemistry. And then proteins are chemistry. So in the, well, you could have an answer that takes 1 hour, or you want a 2-second answer. That is, proteins are important. As I said, I like proteins more than genes. And DNA has only 4 different nucleotides. Protein at least has 22 amino acids. There's more variety. And each protein is all different. Every protein is different. Okay. If that's the answer. I think protein is more important in terms of everything, diseases and functions and so on. Okay. But protein, because I started as a chemist and just become, see, when chemical molecule becomes larger and larger, they become proteins.
Okay, any other questions? Yeah. Maybe I ask one question. Yeah. So you mentioned 3 different methods for study protein structures. Allow the last method, REM, can allow you to directly view the proteins. So would this become overtaking the other 2 methods eventually?
No, I was in. Yeah. It became more dominant, but actually, it's very dangerous to make this kind of prediction because things could turn around. See, scientific development, every 5 years there's a major breakthrough. But particularly right now, what people think is NMR could be sort of phased out, but NMR has its strengths also. Particularly, as I said, we are moving more into dynamics. Yeah. We are now not interested, not so much in static structures. We are interested in how protein moves, or even when it is not moving, nothing is static. It is still vibrating. And NMR has an advantage in that kind of information. So the good thing is 20 years ago, when I met my former colleague, some friend, Professor Michael Chen from CUHK, we were in 2 different camps. We were in the same institute, but he is an X-ray crystallographer. I was an NMR spectroscopist. We don't understand each other, and we couldn't talk to each other. Well, yeah, we collaborated. We had to do a formal collaboration because we were in 2 different fields. But now, both of us, or everyone in the structural biology field, we have to understand everything, all those techniques. In addition, we have to understand biology because technique isn't technique alone that is not useful. Technique have to solve problems. So this is a challenge to the future scientists. Okay, but the future scientists, you may think, well, the old days are easier, but actually, our future scientists. Yeah.
Okay. There's a question over there. There are 2 questions. Yes, please.
Thank you, Professor. I would like to ask, how can we ensure the conformation of the molecules is the same as the conformation inside living cells, like some membrane proteins, for example?
Yeah. Well, how do we ensure the conformation of the molecule is outside is the same as inside? I think we could not ensure. See, in cells, the protein is in an environment that is different from outside. Also, the protein may be interacting with another protein. So you have to, the direction, one of the direction of the future study is to study protein structures in intact cells under in vivo condition, or to study protein structure as a complex of several proteins to get more and more into the real situation. And those studies have been developed, one particular field is called tomography, that is to use cryo-EM to study protein structures inside cells. And that will be developed in the next 5-10 years to become very common.
Is another question? Yes. Yes.
You have mentioned that life science can help us cure disease. Yes, but I would like to ask, maybe will humans live too long if we have too good medicines, and would that be causing a problem? So that be a downside of life science? Thank you.
Well, that is a very good question. I think everything has upside and downside. But when human genome was claimed to be solved in year 2003, the Time magazine claims my generation will live 220. But it's not happening. So it may not be a problem. It's a potential problem, but may not be a problem. But it could increase gradually, but not dramatically, I would say. I think if one disease is cured, another disease could emerge.
Okay. Any questions? Slow question from that side of the audience. So we get one question over there. It's good response. Yeah, we have two questions. In fact, there's another one over there.
Okay. Thank you, Professor, for your really amazing talk. I really appreciate the example about the orchestra because it shows you some creativity inside you because you use this orchestra to let us understand the process happened in our cells, how protein works. So as a scientist, work for so many years, how you treat the importance of creativity, and do you have any things that you do it in practice in your daily life to make sure your mind is fresh full of creativity when you study your projects?
Well, I wish I could have more. Obviously, I'm not creative enough. Otherwise, I may have a Nobel Prize. Everybody, I think this is I think my philosophy is you do your best. That's all. Okay. But I think you need to like what you do. That's important. If you don't like what you do, then you will not be creative. If you like what you do, then you will keep pursuing, keep thinking. Even when you are doing something, then one thing always leads to another. But interaction with people is also important. Okay. I think it's a constant process. And keep your mind fresh is important also. And that means you need to do exercise. You should not like me when I was younger, that I work all the time. I work very long hours, but now I think I need to do more exercise.
Okay. Another question? Yes.
Thank you, Professor Tsai. I know the failure of DNA repair can lead to apoptosis. And what leads to a failure of DNA repair, or is it the speed and is an important factor? Thank you.
Anything. So nothing is absolute in our body. enzyme is not 100% accurate with enzyme. So the DNA replication, even in the best condition, is about 1 million. 1 million. So the error could happen 1 in 1 million. The repair enzyme could be in error also. So there are different reasons. That is, by statistics, things could go wrong when you get old. You know. Or maybe some DNA repair enzyme has mutations. Or maybe your nutrition is imbalanced, then that affects some of the enzymes. So there are just so many factors. And so from the molecular approach, we single out a specific enzyme and study the efficiency. So we could look at every enzyme and study its efficiency and then make a mutation and see what goes wrong, what is the frequency of things going wrong. You could do that kind of study. But in the real life, you know, it's a combination of possible reasons.
Okay, any more questions from the audience? Any more from the internet?